All right, good afternoon. So, as the title indicates, I'm focusing on developing therapeutics for breast cancer. And this is my foray into that field. Uh, and we found some interesting compounds that were acting in a different way than the regular breast cancer therapeutics. And this is an introduction of that class of compounds. So, as we know, breast cancer approximately affects around it's the second largest uh, mortality causing agent for women, and it affects younger and younger women of late than what was seen earlier. It could be because of the improved diagnostics, or it could be some other factors that are driving the occurrence of breast cancer in younger women. And what we see is that at present, the statistics say that 70% of these breast cancers are ER positive, which is estrogen receptor or hormone positive, in other words. And this percentage is actually on the increase with more and more ER positive cases being found of late. And the main therapeutic uh, treatment that is clinically used is the endocrine therapy, where they use tamoxifen and other kinds of agents for treatment. And recently, selective estrogen receptor modulators and degraders have also started being used in the clinical settings. And those have been somewhat good. They have improved the survival chances, but we see that almost 50% of the tumors still exhibit resistance to the current uh, treatment protocols. So, what we see is that there are two main mechanisms that are in play that cause these resistance to current therapeutics. One is the mutations in the ESR1 gene, and the second is the upregulation of mitogenic kinases. So both of these are causing the resistance to be occurring in the breast cancer patients. So we have found a new class of agents that are actually causing the inhibition of growth in mutated breast cancer cells. And these are not acting in the regular sense of estrogen receptor modulation, activity modulation, or degradation, but they are actually acting in terms of mediating the transcriptional repression of estrogen receptor. So we call them, of course, we have this acronym for that. And the compounds are, of course, patented. The methods of use have also been patented. So this is an example of the class of compounds. As you can see, we found several derivatives of the compound that are showing this type of activity. And what we see is that all of the compounds that are shown here have been tested in estrogen withdrawal conditions and also upon treatment with E2 in the presence of E2 also. We see the effect under both conditions. So what this is an example of one of the compounds, and as you can see, these compounds are well characterized with X-ray crystal structure and so on. And what we found is that in organoid systems, we could clearly see that our compounds are actually doing a dose-dependent decrease in the growth of the breast cancer organoids. The main thing was how did we find the mechanism to be transcriptional repression? What we see was that initially, when we looked at the Western blots to look at the levels of the ER alpha, we could clearly see that the ER alpha levels were greatly decreased in the treated cells. And we also found that when we use T47D breast cancer cells, which had the estrogen response element um, fused with the luciferase gene, we found that, in fact, the expression of the luciferase is greatly reduced. So this only showed that ER alpha levels are greatly reduced, and of course the downstream levels of it. And the next thing that we looked at was the mRNA levels for the ER alpha expression. And what we found was that our compound was indeed showing really low mRNA levels, and we could see that different derivatives were acting in a dose a responsive way, where some of them were more potent this was more potent than this other compound in acting in the same way in mediating the repression of transcription. So we then looked at the mutant cells. One of the major issue is the ESR1 mutations are resistant to hormone therapy, and they occur in around 30% of the patients. And the mutations are generally found at Y537 as a serine mutation. And then we also see the second type of mutation, which is D538 as a glycine mutation. And these account for greater than 50% of the mutated ESR1 breast cancers. And the most clinically relevant model here is the Y537S mutation because it's highly metastatic and resistant to most hormone therapies. And it has also shown resistance to the SERMs and SERDs that are clinically being used. So we checked the effect of our compound on these mutated cells, and we found that ours was much better than fulvestrant. The ICI here is fulvestrant. Our compound was much better than fulvestrant in inhibiting the growth of the Y537S cells. And in fact, we could also see the lowering of ER alpha levels and also the lowering of the downstream proteins, which is the PGR and the GREB1 proteins. So this shows that not only is the ER alpha levels brought down, but other responding protein levels are also brought down by these agents. And we found the same kind of growth inhibition in D538 cells, and ours was indeed, some of them were as good as fulvestrant, but some of them were better than fulvestrant in this particular study. So we have done initial studies in mice. The compounds seem to be well tolerated, and they seem to be inhibiting the growth of the breast cancer tumors. But we are repeating those studies at present, so we hope to have more results on this going forward. So the main thing is the mechanism of how this is happening. We do feel that it could be some kinases, but so far all the kinases that we have investigated that are being inhibited by these compounds do not seem to be having an effect on the transcription repression. So we are trying to assess how else it is happening, and we can clearly see that it is not on the promoter where estrogen is sitting, because these mutations are essentially on that promoter, which causes the estrogen to be constitutively active. So it's not on that promoter, it could be on some other promoter, and that is what we are investigating. And of course, this was not done alone by me. I'm only a chemist. I have several biologists who are on the team. So Dr. Chris Williams, Dr. Matt Burrow from Tulane, and Dr. Sridhar have been working on the biology aspect of it. And Dr. Komati was the person who synthesized many of these compounds. And Dr. Kim is another biologist who is working on it. And as you all know, Xavier is an undergraduate institution, so I have several undergraduate students performing the synthesis and doing the bioassays, and you can see several of them named here. I would like to thank my funding agencies. It's all done because of the LCRC Bridge Grant. Many of you know what LCRC is. And of course, the NIH Build Grant helped in funding the undergraduate students. So I will take questions at this time. Thank you.
So one of the wackadoo things about SERMs is that they kind of act differently depending on tissue type. They could be
Yes.
agonist in one area and antagonist in a
Antagonist.
completely different one. Do you have that same issue have you looked at other tissue types? Do they act differently? I can't remember your acronym. Is it MERS?
Mirrors. Yeah.
Okay.
Mirrors.
Good remember.
Yeah. So far, we have been looking at breast tissues, but they do act differently. For example, we know that many of the other cancers are also estrogen dependent, like ovarian cancer and prostate cancers and others. But mirrors don't act on them. So, but they do act on other types of cancers, like leukemia. It seems to be having a great effect. So we do think the mechanism is very, very different.
Have you looked at the role of E3 ligases and or these compounds functioning as molecular glues?
Tell me again. Sorry. E3
So
ligases?
the core structure you have usually functions at least in terms of things like that recruits E3 ligases, and then functions also in the case of the degradation that you're seeing of the estrogen receptor could be the result that you're actually recruiting an E3 ligase, which is actually then
Yeah, so far we have
removing the estrogen.
investigated that, and that doesn't seem to be the case.
All right, thank you.
We still are not clear on the mechanism of it. Thank you.